Good afternoon, everyone. My name is Siphumle Jama. I'm a PhD student from Stellenbosch University in the Department of Horticultural Science. Today, I'm going to be presenting our research, which is titled Gas Diffusion during storage and its relation to internal disorders that are found in palm fruit. And it's under the supervision of Dr. Elke Crouch and Dr. Alemaye Utsike, and also our collaborators from Leuven. While storing fruit under CA, or rather low oxygen conditions, is very good in terms of extending the long-term storage quality of fruit, but also this can lead to the development of physiological internal disorders, such as internal browning. So this can lead to a shift in respiration from normal respiration to fermentation, which can lead to the development of ethanols instead of just normal energy, and this can result to internal browning in fruit. So the aim of this project is to understand rather how does gas diffusion play a role in terms of the development of physiological disorders. In us actually trying to find better storage protocols, we are aiming to measure respiration rates in terms of oxygen intake and also carbon dioxide production, and also make use of the microstructure of the fruit, as that is the main channel of gas during storage, and also develop a gas diffusion model that we perhaps can use in order to make prediction in terms of which CA storage regimes lead to the physiological disorders. So this is the model that we are planning to use. So just to go quickly through the methodology in terms of us measuring our respiration, we are going to look at 3 different palm cultivars, which is Forel, Fuji, and Rosy Glow. And also we're going to look at inside and also outside fruit from the canopy position. And also these are some of the 2 storage regimes that we are using, which is storage 1 and also storage 2. So there are 3 students that have actually conducted research. I'm the 4th one, so I'm just going to go briefly through the summary of the 3 theses, which is what we found in terms of the respiration rates. Forel is one of the cultivars that is very sensitive and is showing high respiration rates as compared to the other 2 cultivars from both storage 1 and also storage 2. And also, it's just linking that into the IB that they actually found. We saw that from 2023, Forel was the fruit that showed the highest browning from both storages 1 and also storage 2. And also, which is a new addition to the work, which is making use of CFD modeling in terms of making predictions as what it can do for us. So it's very important in terms of measuring the respiration quotient, which is RQ, which is a very good indication of the internal browning that we notice. So if you're going to look at highlighted C under 0.05% of oxygen, where if you look at the fruit that is cycled red, there is a very intense red color in the middle, which mainly highlights the onset of fermentation. So that is what we are trying to be able to predict with using modeling in this study. So in just summary of what they found actually was that most internal browning is found in Forel fruit, and also secondly Rosy Glow, and also the outside fruit is one of the canopy positions that is affected the most. And also what is most importantly is the tissue structure. If you would observe it, the more healthy fruit, the porosity is rather more clear, and which actually means that the gas diffusion is much more efficient and flows very nicely. And from the IB affected tissue microstructure, the porosity of the fruit is more flat, and that actually affects the flow of the gas transport during CA, and that leads to the internal browning. So my PhD and also addition to the work is actually combining everything into a summary, and how can we use our CFD modeling and a gas diffusion model in order for us to make predictions as to which storage conditions under CA using different storage regimes lead to the most browning. And in my approach is that we're going to make use of CT analysis, where we're going to be looking at the microstructure, which is the porosity. Those are the main channels of which gas diffuse into the fruit. and also look at respiration as well. It is the rate that these gases are traveling through these pores and how we can bring that together in terms of developing this model, and also how can we make use of this model to make future predictions. We can use any value when the model is development or any storage. We don't have to run the normal CA, but we can make prediction in order for us to know which storage kind of give us the most internal browning during storage. Thank you.